Good morning, good afternoon, and good evening. Once again, welcome to my channel. My name is Delvin Wenani, and today I want to take you through my first prediction for 2025 KCSE Biology Paper 1. I'll be taking through some of these predictions, and these are not necessarily predictions before the KCSE, but these are just revision questions that have a format of the KCSE. So sit back, relax, share my channel, and view up to the end. And if possible, leave us a comment, and we shall be appreciating. More predictions will be coming, and this is my first prediction, first revision in 2025. So welcome to my first prediction revision biology paper 1. Now, the first question. Give a reason or reasons for the following precautions during collection and after study of specimens. Now, the first precautions we're being asked is why, question number A, returning specimen to their natural habitat. Remember, we are always advised that we collect the specific number, and number two, we also need to make sure that we return the specimens to their natural habitat. Why? And this is basically to maintain an ecological balance. If you distort the ecology, the balance may be distorted, and therefore, if you are using a specimen, specifically the live ones, you're using them maybe for experimental purposes, you are supposed to maintain them, to keep them safe, and return them so that we can maintain an ecological balance. Then B, not destroying the specimen. Why do we always say that you should not kill the specimen? And the reason is because to avoid distortion of specimen features. You can take maybe, for example, a grasshopper, and then you remove the legs. And you know, grasshoppers has legs specifically for jumping, specifically for moving. So you end up destroying and distorting the specimen's features. Then question number 2, list down the reasons for the following importance of carrying out classification of living things. So number 1, why do we put, that is not number 1, but number 2A, putting living organism in the correct group? When we are doing classification, why do we put living organisms in the correct group? And this is for reference. It is for reference. Then B, why do we arrange, that is the question is asking, arranging information about living things in an orderly manner? Why should we arrange in an orderly manner? And this is to avoid chaos and confusion. Then, number 3, state 2 differences between modern and artificial classification. Now, there are 2 types of classification we are talking about here. There's the modern classification, and then there's artificial classification. And this is some of the differences. 1, in modern classification, it is known worldwide. But artificial classification is a local one. The classification that you do it on a local, maybe a local venture, a local place. Number 2, modern consists of 2 part names. We call them the generic and specific names. It has a name which consists of 2 parts. That's a generic and specific name. But when you come to artificial, a name is not split, just a name. Then a modern, another difference between a modern and artificial is modern is based on phylogenetic, while artificial is based on superficial features. It's based on superficial features. Then state 3, that's question number 4. State 3, distinguishing characteristics of bacteria. State 3, distinguishing characteristics of bacteria. Number 1 is their cell wall is made of membrane. Then they have very few organelles. And then number 3, about the bacteria, they are prokaryotic. Those are the 3 distinguishing characteristics of bacteria. Number 5, we are being asked, enumerate 3 ways of controlling spread of epidemic diseases. Enumerate 3 ways of controlling spread of epidemic diseases. Number 1, the common one that we used 4 years ago is quarantine. Quarantine. Number 2, we can also use isolation. And then number 3, we can use vaccination. Those 3, quarantine, isolation, or vaccination. Question number 6. Explain how the following factors influence distribution of organisms in an ecosystem. Explain how the following factors influence distribution of organisms in an ecosystem. Roman 1, change in pH. That's change in pH in soil. So when there is low pH, there will be leaching acid. That's what we call low pH leads to leaching acid. So nitrogen fixing organisms will be denatured. Then number 2, the saprophytes will also be interfered. Then question Roman 2, when there is strong wind in Masai Mara National Park, what will happen if there is strong wind in Masai Mara National Park? 1, it will destroy trees, and then it will also lead to high rate of transpiration, and it also bring about rain-bearing clouds. It will bring about rain-bearing clouds. State 2, behavioral adaptation of lion, which makes it efficient for predation. We're talking about behavioral adaptations of lions, which make it efficient for predation. So number 1, this is how lions behave. Number 1, they move against wind direction. And why do they move against wind direction? So that they can get the scent from the prey. Number 2, the lions, they camouflage to hide themselves from prey. Then number 3, they also produce sound similar to prey to attract the prey. Producing a sound similar to the prey to attract the prey. Question number 8. Question number 8. Name the parasite that causes amoebic dysentery. Name the parasite that causes amoebic dysentery. Now, amoebic dysentery is caused by a parasite by the name Entamoeba histolytica. It's caused by a parasite by the name Entamoeba histolytica. That's a scientific name. So when you're writing, make sure if it is handwritten, make sure it is written separate and underlined separately. The first name, which is a generic name, should start with a capital letter, and then the second name, with a specific name, should start with a small letter. Question number 9. State any 3 effects of soil. State 3, take again. State 3 effects of oil spillage in aquatic environment. State 3 effects of oil spillage in aquatic environment. So number 1, the causes of oil spillage, it cuts off oxygen and carbon dioxide supply. And this one will affect the aquatic plants, and also it will affect the aquatic animals. Remember, inside the water or the aquatic environment, we have living organisms, which we call the aquatic living organisms. Those are plants and animals, and they require oxygen for the animals and carbon dioxide for the plants. Number 2, it reduces light penetration that is required during photosynthesis. It reduces light penetration that is required during photosynthesis. Number 3, it clogs birds' feathers, making flight difficult. It clogs birds' feathers, making flight difficult. Then number 4, I can give an extra. They asked for 3, but we can give 4. An extra one, it blocks the respiratory surfaces of organisms. It blocks the respiratory surfaces of organisms. Now, the organisms I'm talking about here, I'm talking about the aquatic organisms. Those are plants and animals. And most of the aquatic animals, they have gills. So the oil will clog the gills, block the respiratory surface, and therefore the organism will not be able to exchange gases. Question number 10. The concentration of carbon dioxide in a tropical forest was measured for 24 hours. The graph below shows the results obtained. Now look at that graph. Look at that graph. Then let us respond to the question. The question is asking us to account for the results obtained at A, midnight. Midnight. Now when you look at midnight, discover that there is very low light intensity, and therefore there will be no use of carbon dioxide in the process of photosynthesis. Once there is low light intensity, there will be no use of carbon dioxide in the process of photosynthesis, and therefore the amount of carbon dioxide will be high. The amount of carbon dioxide will be high. Then at noon, that is during the day, that's question number B, at noon, there is high light intensity, and therefore there is maximum use of carbon dioxide for the process of photosynthesis, and therefore the amount of carbon dioxide is less, very low. Why? Because it's being used in the process of photosynthesis. Now, question 11, name the respiratory surfaces of the following. Name the respiratory surfaces of the following. The first one is mosquito larvae. A mosquito larvae. So a mosquito larvae uses the tracheoles. It uses the tracheoles for gaseous exchange. A respiratory surface is a surface that is used for exchange of respiratory gases. Then number B is a tadpole. A tadpole uses gill filaments. It uses gill filaments. Question number 12A. What name is given to the reaction of the body after coming into contact with a pollen grain? Take again. What name is given to the reaction of the body after coming into contact with a pollen grain? And that one we call it an allergy, or we call it an allergic reaction. You can say it's an allergy or an allergic reaction. So basically an allergy or an allergic reaction is, in the common term we say it is a hypersensitive reaction. A reaction of the body beyond the expectation. So it becomes hyper. Hyper means overreaction. Question number B. Explain how anaphylaxis occurs. Explain how anaphylaxis occurs. Now, this is where an antigen will lead to overproduction of antibodies. Remember, an antigen, any foreign substance entering your body is termed as antigen, and therefore the body reacts by producing antibodies. And these antibodies will come to react, or will be produced to react against the antigen. Now, this antibody-antigen reaction will now lead to production of histamine. Now, the histamine will now cause overdilation of blood vessels. And when the blood vessels are overdilated, we shall say you are suffering from anaphylaxis. Question number 13. The diagram below shows the tracheal system of an insect. Study it and use it to answer the question that follows. Take it again. The diagram below shows the tracheal system of an insect. Study it and use it to answer the question that follows. Question number A. Name the structure label X. Name the structure label X. Let us look at the structure label X. When you look at that, that one is a spiracle. That one is a spiracle. Question number B. State how the structure X is adapted to its function. State how the structure label X is adapted to its function. Now, first of all, we agree that this part label or the structure label X is a spiracle. And therefore, if it's a spiracle, then it must be having hairy. Most of the spiracles are hairy. And this is to trap solid and also to prevent solid particles from entering the system. It also has what we call muscular valves. And this controls the opening and closing. It has muscular valves. Question number C. How is ventilation maintained in trachea system? How is ventilation maintained? Now, in all the trachea systems, they have what we call chitin or chitin. Now, chitin is one that keeps it open. And in another language, we can say it prevents it from collapsing. So it keeps the trachea system always open. Question number 14. List down 2 processes that transport manufactured food in plants. List down 2 processes that transport manufactured food in plants. So how are manufactured food transported in plants? Manufactured food, they are transported through 1, active transport. They can also be transported through diffusion, or the common one is what we call mass flow. They can also be transported through mass flow. Question number 15. Describe the function of insulin in the human body. Describe the function of insulin in the human body. Now, insulin, the major function of insulin is actually to stimulate the liver cells to convert excess glucose to glycogen for storage. Remember, it's the liver cells that will convert the excess glucose to glycogen for storage. So, insulin simply stimulates the liver cells. Number 16, define homeostasis. Define the term homeostasis. Now, homeostasis simply means maintaining a constant internal environment despite the changes in the external and internal environments. For example, maintaining temperature at 37 degrees in humans, despite the temperatures outside being either high or low. That we call it homeostasis. Maintenance of a constant internal environment despite changes in the external and internal environments. Question number 17. Distinguish between positive and negative feedback as used in homeostasis. Distinguish between positive and negative feedbacks as used in homeostasis. Now let us start with the negative feedback. Now, negative feedback is a mechanism that restores, or what we call it, corrects a worsening situation. For example, if the level of blood sugar in your system is high, your system will try to lower the blood sugar. That's a negative feedback. But a positive feedback worsens the situation. If the sugar level is high, it makes it to be higher. That is called positive feedback. So negative feedback restores or corrects a worsening situation, whereas positive feedback worsens the situation. Question number 18. Premature baldness is due to a recessive gene located on one of the sex chromosomes. We are being asked to construct a genetic cross of marriage between a husband who has a premature baldness. Now, we are using letter small h to represent the gene for premature baldness. Using letter H to represent the gene for premature baldness. So that is the cross. Look at that. You can pause, then do your cross, then compare with that. Remember, the premature baldness is located on one of the sex chromosome. And because it's located on one of the sex chromosome, then definitely it will affect either of the two. But we are coming to that. So we are crossing a cross of marriage where the husband has premature baldness. So question number A, they're being asked, explain why most of the colorblind people are male. Explain why most of the colorblind people are male. Now, colorblind can affect both male and female. It is sex-linked, but colorblind is linked to the X chromosome, not Y. Now, maybe I can go back to the other question that I was talking about, the baldness. Now, premature baldness, the gene that controls premature baldness is located on the Y chromosome. And that's why it's only the male that have what we call the premature baldness. It's not in female. Why? Because it's located on the Y chromosome. And it's only the male that inherits the Y chromosome. But now when we come to color blindness, it will affect both female and male. But the question is asking why most of the colorblind people are male. Now, I want to say this. Most of the male are colorblind because male inherit only one X. Because X chromosome the color blindness gene is located on the X chromosome. Male have one X, one Y. And therefore, for a man, they inherit one of the X's. While the female, they inherit two X's. So for a female to be colorblind, the female requires both the X's to be affected. What I mean is the two X's in the female, all of them must be having the gene for blindness for a woman to be colorblind. But for a man, because they are inheriting only one X chromosome, if that X chromosome is affected, then that man automatically becomes colorblind. Okay, so the reason why majority of the male are colorblind is because they inherit only one X chromosome. Question number 19, explain how surface area to volume ratio affects the rate of diffusion. Explain how surface area to volume ratio affects rate of diffusion. Now, an organism which has a large surface area to the volume ratio, that organism will definitely have a high diffusion rate. And this is because the molecules pass through a very short distance. The molecules pass through a very short distance. Question number 20. Distinguish between osmosis and active transport. Distinguish between osmosis and active transport. Now, I will start. Now, the question is asking us to distinguish. The question is not asking us to differentiate. We need to get the differences. Now, maybe I can state those ones. When you are being asked to distinguish, it means if you get one of the terms correct, for example, if you get osmosis correct and you fail to get active transport, you will be awarded a mark for that. But if you are asked to differentiate, then it means the two answers are binded together. That means for you to score, you must give clear definitions for osmosis and clear definition for active transport for you to score. Now, for this case, we are being asked to distinguish. So we give differences separately. We can give osmosis marked separately, then for active transport marked separately. So let me start with osmosis. Now, osmosis is the movement of water from their region of high water concentration to a region of low water concentration across a semi-permeable membrane. It's the movement of water molecules from a region of high water concentration to a region of low water concentration through or across a semi-permeable membrane. While active transport involves movement of particles. Now, look at this. In osmosis, you're talking about movement of water molecules. Here, you're talking about movement of particles from a region of low to high concentration across a semi-permeable membrane, but energy is required. Now, for the process of active transport, energy is required. Question number 21. The diagram below illustrates an experiment on respiration in a small insect. The diagram below illustrates an experiment on respiration in a small insect. Let's look at the diagram. Look at it very carefully. Then the question they're asking is, explain the observation made after 10 minutes in the above experiment. Explain the observation made after 10 minutes in the above experiment. Now, if you are being asked to explain the observation made, the first mark will be at the point where you give or you state what you are seeing. First of all, you must tell us what you are seeing before explaining. You are explaining the observation made. So give us first of all the observation, then give us the explanation. So for this case, when you look at the diagram, the first mark is there is a rise in the level of colored water in the delivery tube. That's a mark. We can see there is a rise. So by just stating that there is a rise in the level of colored water in the delivery tube, that's a mark. Then now you can proceed and give the reason or your explanation. Now the explanation is because the insect is respiring. And when the insect is respiring, it is using oxygen. And when it is using oxygen in the flask, then remember also during the respiration, as it's using oxygen, it is releasing what? Carbon dioxide. Carbon dioxide, because of the presence of sodium hydroxide, it will be absorbed by sodium hydroxide. So sodium hydroxide will absorb the carbon dioxide, which is being released by the respiring insect. And this one will create a vacuum, which will reduce in pressure. And because now the outer atmospheric pressure is high, it will now press or push the level of water. So atmospheric pressure will press will push the colored water, and the colored water will rise because the outer atmospheric pressure is still high. Question number 22. The diagram below shows the internal structure of a leaf obtained from a plant in a certain habitat. The diagram below shows the internal structure of a leaf obtained from a plant in a certain habitat. Now, different plant structures have different adaptation depending on where they are grown. Different habitats have different structural adaptation of their plants. This is what I mean. We don't expect a plant which is grown in what we call an aquatic environment to have the same structures as a plant which is grown maybe in a terrestrial or which is grown in maybe an arid and semi-arid areas. We don't expect them to have the same structures. And therefore, in this case, we're being asked, state two observation or what we call the observable adaptations of the leaf to its habitat in relation to gaseous exchange. They're asking us to state two observable adaptations of the leaf to its habitat in relation to gaseous exchange. Now, when you look at that structure, there is clear, you can see air spaces. Now, if you see air spaces in a leaf, anytime you see air spaces in an organism, it means it's something to do with buoyancy. Buoyancy is the ability to float. So it means this plant or this leaf must have been from a plant which is an aquatic plant. So there's air spaces for storage of air for buoyancy to enable this leaf to float on water. And as it's floating, it can also carry out what? Respiration. Then you can also look at that. When you look at that structure very well, look at the cuticles. The cuticles are very thin. Now, when you see thin cuticles, thin, sometimes even they lack cuticles, thin or lack cuticles, then it means this plant must be in an environment where there's a lot of water, and therefore it is trying to reduce the size of the cuticle so that to reduce the distance for diffusion of those gases, and at the same time also to increase or to expose more stomatas for gaseous exchange. So those are the two things we are supposed to look there. One, we are seeing air spaces, and I've said air spaces are for buoyancy. Then number two, we can also look at thin, it's almost lacking, thin, lacking cuticles. So, and this is to reduce the distance for diffusion of respiratory gases. We can also, if you look at that diagram very well, you can also see there is large, the stomatas are very large. And the reason why the stomatas are very large is to do what? To increase surface area for gaseous exchange. The stomatas are quite large to increase surface area for gaseous exchange. Those 3 points. Number one, air spaces for buoyancy. Number two, thin cuticle to reduce distance for diffusion. And then number three, large stomatas to increase surface area for gaseous exchange. Now, apart from question number, that is Roman number 2, apart from stomata or stoma, name other sites for gaseous exchange in plants. And these are 1, lenticels, and we also have what we call cuticles. Now, cuticles are mainly used in what we call young epidermis. Cuticles are in young epidermis. Now, Roman 3, state 2 adaptation of spongy mesophyll cells to gaseous exchange. State 2 adaptation of spongy mesophyll cells to gaseous exchange. Now, how this now question is out of the diagram, but it's an adaptation question. So we are being asked to state 2 adaptation of the spongy mesophyll cells to gaseous exchange. Now, we all know that spongy mesophyll cells have a thin wall. Their walls are very thin. Now, the reason why they have a thin wall is to reduce the gaseous exchange distance. To reduce the gaseous exchange distance. Now, if you also look at most of the spongy mesophylls, they are moist. They have what we call a moist membrane. And for any respiratory surface that is moist, actually, the reason why most of the respiratory surfaces are moist is to dissolve what? Respiratory gases. Now, most of these respiratory gases, they are transported when they are in what? In liquid form. So they must dissolve. So that's why most of the lungs, look at the lungs, they are moist. Why? To dissolve the respiratory gases. That is oxygen and carbon dioxide. Oxygen and carbon dioxide, which are respiratory gases, they are transported in a dissolved form. So they must be dissolved. So if you are asked why it is moist, definitely is to dissolve respiratory gases. Question number 23. Distinguish between guttation and transpiration. Guttation and transpiration. Very fast. We are now distinguishing also. Guttation is loss of water in form of water droplets. You can say that. But you can also say through hythadotes. If you say it is a loss of water through hythadotes, you also get a mark for that. But transpiration is loss of water in form of water vapor or loss of water through stomata, lenticels, etc. So when you use transportation of water, loss of water in form of water vapor, that's correct for transpiration. You can also say loss of water through the stomata or the lenticels, you also get. Now, because they have not stated, they are only asking you to distinguish. So if you're being asked to distinguish, you can either give how it does, or what it is. Yeah, and that's why we are saying if you're being asked to distinguish between guttation, you can say it's a loss of water through hythadotes. You get it. And then transpiration is the loss of water through stomata. That's correct, because we have given the distinguishing. They are not specific. They have not said define. No, they have not said you define. They have only said give differences. So I can decide to say to give differences depending on where you occur and where you occur. And someone else can give a difference based on who are you and who are you. Loss of water in form of droplets, that's guttation, and loss of water in form of water vapor, that's transpiration. All of them correct. Question number 24. Name the tissue in plants responsible for transportation of carbohydrates. How are carbohydrates transported, or where are they transported? In plants, carbohydrates are transported through phloem. Phloem tissues. They're transported through phloem tissues. Mineral ions are transported through xylem tissues. Mineral ions are transported through xylem tissues. Question number 25. The experiment setup shown below investigates a certain physiological process in plants. Use it to answer the question below it. Now that is the experiment, and this experiment is set up to investigate a certain physiological process in plant. Now, the first question we are being asked, what was being investigated? When you look at that experiment, definitely this one they are testing for what? Transpiration. This one they're testing for transpiration. Then the question B, give the role of oil layer in the experiment. Give the role of oil layer in the experiment. Now, oil, majorly if you see oil is for prevention of gases, but in this case, the oil layer is used to prevent evaporation of water from the beaker. Oil layer in this case is used to prevent evaporation of water from the beaker. Question number 26. Define mutation. Define mutation. So what are mutations? Mutations, we say they are sudden. We can use the word sudden. We can use the word rapid. We can use the word spontaneous. These are changes that occur without giving a notice. So these are sudden, rapid, spontaneous changes in the genetic makeup of an organism. We call them mutation. Sudden, rapid, spontaneous changes in a genetic makeup of an organism. Question number 27. Question number 27. The diagram below illustrates some of the beaks in birds. The diagram below illustrates some of the beaks in birds. Look at those beaks. Question number A. Identify the evolutionary phenomenon displayed by the beaks above. Identify the evolutionary phenomenon displayed by the beaks above. That is adaptive radiation, or you can simply say divergent evolution. Divergent evolution. Now, when I demonstrate by the hand, divergent, it means we from the same diverge. Divergent evolution. And I'm going to explain in Roman B. They're asking, explain how the phenomenon above might have risen. Explain how the phenomenon above might have risen. Now, divergent, same embryonic origin, but adapted to perform different functions. That's what we call divergent evolution. Now, we have the same embryonic origin. When you look at the beaks, all of them, they are originating at the same point. But at the end, when you look at the beaks at the end, each one of them is adapted for different functions. Some are for sucking honey, others sucking nectar, others are for fishing, others for cracking nuts. So, same embryonic origin, different function. Why? Why do you think they are diverging? And this might be because they are exposed to what? Different habitats or environment. They are the same, yes, but we are exposed to different habitats. So you find yourself in a habitat where maybe your only way to survive is by fishing. So you adapt yourself to fishing. Or maybe the only way you survive is by cracking nuts. The only food that is there is nuts. So you must get adapted to cracking the nuts. So the birds might have been exposed to different habitats or environment, and therefore their beaks became modified to enable them to feed to the food available on the environment. To enable them feed on to the food available onto the environment. Question number 28. List any two limitations of fossil record as evidence of organic evolution. Now we use fossil record as an evidence of organic evolution, but it has limitations. Number 1, missing links. Missing links. Number 2, lack of fossilization. Number 3, distortion by what we call geological processes. Then number 4, which is very common, scavenging of the animal material. Scavengers. Then number 4, scavenging of animal material. The scavengers, they feed on those fossils, and therefore they cannot be found. Question number 29. Explain why Lamarck's theory of evolution is disputed by modern scientists. Explain why Lamarck's theory of evolution is disputed by modern scientists. Now, Lamarck's theory bases on the transmission of characteristics. That the characteristics that you obtain, you transmit them. So, phenotypically acquired characteristics, like going to the gym. If you go to the gym, you gain muscles, Lamarck says those muscles shall be transmitted to the child. So, scientifically, we tend to refuse that theory. Why? Because phenotypically acquired characteristics cannot affect the genotype of an organism. Phenotypically acquired characteristics cannot affect the genotype of an organism. When I say phenotypically acquired, I'm talking about the physically acquired, your appearance, maybe height, maybe thickness, etc. Those ones are phenotypically acquired characteristics, and they cannot be, or they cannot affect the genotypic of an organism. Question number 30. List 2 hormones that stimulate germination. Now, germination is stimulated by hormones, and these hormones are gibberellins, ethylene, and even cytokinins. They have given us to name 2, but even cytokinins stimulates germination. So 3 hormones: gibberellins, ethylene, and even cytokinins. Number 31. Name 2 parts in a cell where respiration occurs. Name 2 parts in a cell where respiration occurs. Respiration in a cell occurs in the matrix, that is the liquid part of the mitochondria, and also it occurs in the cytoplasm. They will ask you this question to give maybe a difference between respiration and photosynthesis. Respiration is only process that occurs in 2 cell parts. It occurs in the mitochondrion, that is matrix of mitochondria, and even the cytoplasm of the cell. Stage 2, structural adaptation of anemophilous. We call them the anemophilous flowers. Stage 2, structural adaptation of anemophilous flowers. That's question number 32. Now, many pollen grains. This is an adaptation. Many pollen grains to increase chances of pollination. It also, the anthers are losing, or they are loosely hanging, exposed to the environment, so as the pollen grains can be easily transported. Last question, question number 33. State the function of the following in human digestion. A, cholecystokinin. Now, cholecystokinin is a hormone which is produced in the duodenum. Once food leaves the stomach and enters the duodenum, there are 2 hormones that will be produced. Cholecystokinin, the one we are being asked, and there's another one we have not been asked, but it will also be reduced, known as secretin. So there is cholecystokinin and secretin. Now for this case, they're asking us, what is the function of cholecystokinin? Cholecystokinin stimulates the pancreas to secrete pancreatic enzyme, or what we call the pancreatic juice. Now, it also stimulates the gallbladder to secrete bile. Stimulates the gallbladder to secrete bile. Question number B. They're asking us to state the function of smooth muscles in the stomach walls. The function of smooth muscles in the stomach walls. Now, the stomach walls have muscles, and the major function of those muscles is contracting and relaxing to bring about churning of food. Once food enters the stomach, it's supposed to be churned, churning, turning in simple terms. So who does that? Smooth muscles in the stomach walls. That one brings us to the end of our first revision. Remember to share, remember to subscribe, remember to follow, remember to leave a comment for us so that we can know where to improve. Otherwise, God bless you until we meet again. Bye.